So we're going to start chapter 1. And it looks something like this. Okay, so let's dig in. Let's get ourselves set up. So chapter 1, obviously there's in chapter 1 going to be a lot of ideas, kind of general broad concepts to kind of pull biology together. One of the things I also like to do is take a little bit of time, perhaps maybe even, dare I say, too much time, but well intentioned time, to kind of lay down something that probably not a lot of biology instructors really do. And that's kind of to spend a couple of slides early on, kind of forming a little bit some of the kind of the philosophical underpinnings, kind of the ways of thinking of science in general. Because a lot of times that helps, because a lot of times what we do is we just start plowing through the content and we're just like a runaway freight train, and you guys are kind of like white knuckled hanging on for dear life. And nobody, especially us, really stops to consider that the average person probably doesn't think about this stuff as much as we do. And it goes without saying that when you're looking at science in particular, there is kind of a way of thinking that goes into it, right? So when you're thinking scientifically about scientific things, it's a different way of thinking about things and a different way of going about things than, say, if you were reared in like the humanities or things of that nature, or philosophy of that, or something of that nature. It's very distinct, has a very peculiar signature to it, and there's some hallmarks to it. And one of the reasons also why I want to do that is because in the first week in particular, right, I'm always really big about kind of full disclosure, the kind of orientation sense of like, okay, so this is kind of who I am. This is what makes me tick. This is kind of how I see things. We started that process in the lab when I got all mush-headed on you guys. But now more intellectually, I like to do the same thing so that you can kind of see the direction that I come from when I approach science. Okay, and it's not good or bad. It's just kind of the way the content is flavored. Every instructor has kind of their filter that things go through, and oftentimes it'll shade. We see that as sort of nuances or emphases, perhaps, that we see from one instructor to another. But it all kind of gets informed through that filter. And I want to talk a little bit about that filter, kind of the way I see the discipline itself, because that's what we're doing. We're introducing ourselves to the discipline of biology specifically, but science sort of in general. And there's a couple of really important things and really important messages that I want to start now, and that I typically hit on and touch on and reinforce as we go through the semester. Okay, so let's start off with the concept of diversity, right? So first of all, what I want to do is I want to first of all we're going to talk about diversity, right? This is our first big biological concept, right? This is the revelation that we had not so long ago, actually, in Darwin's time, actually, of just how much stuff was out there. And not only that, but like how many different things were out there as well, right? So we didn't really have a good understanding of just all the things that are out there until the 19th century. Now, for philosophical purposes and to kind of pin down some of the way I see things is I am an unapologetic, this is kind of a theme, right? So that you've already picked up on in the first day, right? I am what I am and I don't apologize for it. But I tend to be, probably more so than others, a little bit of a purist when it comes to science, a scientific purist, which means that when I see the world, I see it scientifically. When I've got my scientific lens on, it's got limits, it's got parameters, and it is pure, right? And a lot of times people don't have that. And when you're looking at it, it's actually kind of bundled into this concept of sort of the ways of thinking about things. So some of you guys have already been encountering that. There are intellectual disciplines that you already have encountered, right? How many of you guys have thought about philosophy or read anything philosophical in your life? That is a field of thought. That is an intellectual discipline that has its goals and its purposes and its intentions, right? Some of you may have kind of gotten interested into, perhaps or not, an offshoot of philosophy, theology, right? So something that has more of like a religious tone to it, a completely different set of goals aside from philosophy or whatnot, right? But it's got its focus, it's got its parameters, and it's got its targets, right? So it's got a very definable purpose. Science is a third field of thought, and probably the youngest, right? It's kind of the youngest brother of the trio. But it, unlike the others, has the most distinction and the most wired design, and some would argue the most limitations, okay? Because it is very specifically designed to do very specific things. And when it gets outside of those designs and those intentions, it becomes very dysfunctional. That's the reason why I'm a purist, because when it's doing its job, there's nothing like it. We have nothing like it. But when it's outside of its job, it becomes very dysfunctional and very abstract and very difficult to wrap our heads around. So here's the reason why I say that, because as a scientific purist, there are two things in science in particular that permeates all the way throughout biology, and we'll see this throughout the semester. And we'll see this also later on in the chapter, but I want to lay them down now. Unlike the other two disciplines, science has two requirements before you even enter the room. Number one is it requires objectivity, right? There is absolutely no alternative. Non-objectivity is not science. It's philosophy or theology or whatever you're talking about, right? So it's not science. It requires objectivity. That's a requirement of science. The second one is testability. Unlike the other two, which don't require testability at all, science requires it. And that really defines science in a very, very kind of a much narrower set of parameters than the other two. Now, the reason why I say that is because when I look at biology in particular as sort of the manifestation of science studying life, which is what we're doing, biology in a lot of ways is very much like a very huge, very complicated, but beautiful tapestry. And if you've ever looked up close to a tapestry, one thing you'll notice is that a good tapestry is composed of many threads. Many concepts and many ideas weave our biological understanding together. But if you look at a good tapestry also, no thread dominates another. They all work together in balance. And when they all work together in balance, then you see the beautiful picture that comes out in the tapestry. One of my big things is balance. A lot of times, because people study biology, we also have the same flaws that people have. We are egomaniacs. We get a little big of ourselves, right? And a lot of times we're delusional. I would argue that researchers are, for the most part, powerfully dysfunctional people who are at the same time brilliant. Notice I started off with dysfunction first, right? But the thing with biology is there's a lot of concepts we're going to cover in biology. But one thing I always want you to remember is that all these concepts, there is no favorite son here or daughter, right? They're all together in balance, woven together perfectly, okay? So they're all in balance. And one of the reasons why I say that is because the one thread that oftentimes gets out of balance is this guy right here, right? How many of you guys are familiar with Dawkins' famous quote? Right? Nothing in biology makes sense unless viewed through the lens of evolution. Which I would argue is actually wrong, because there's a lot of other things that factor into and weave with evolution that also are important threads in order to understand biology. And I'll explain that here in just a second. So one of the things that I always like to sort of throw out there is every instructor is going to have their thing, right? Their bias, their sort of filter. Mine is genetics. So actually, I would rephrase Dawkins' famous quote into something a little more, I believe, accurate, which is nothing in biology makes sense unless viewed through the lens of genetics. I'll explain why. And I'll try to make my case here in just a second. And I'll continue, by the way, making that case. But this is the thing, right? So oftentimes we give so much energy to evolution that we kind of get this very powerfully distorted view, right? That all these other things, all these other ideas, all these other concepts are non-existent or in servitude to one idea, when in reality they are all peers and they're all woven together, right? And there's a pet peeve, I admit, there is a pet peeve that I have, because a lot of times what you see in a textbook on evolution is so shallow and so thin that it's almost better actually not to say it than to say it so shallowly, because there's so much going on in the idea of evolution. There are so many things that weave into. There's so many complexities, so many facets, that you kind of are being a little dishonest, I would think, intellectually dishonest, to make it sound so simple, because it's not. And there's a lot of great stuff in it, right? But like any other idea that we're working on biology, I kind of want to sort of demythologize evolution. I want to sort of how should we say, theologize evolution, right? So evolution is scientific, not theological. And as a result, we're still working on it, right? So it's a scientific idea. Some parts of evolution are very strong and robust, and we feel pretty good about it. There's other parts that we're still kind of not quite sure how it all came together. You know, we're still kind of trying to put our ideas together. We're still trying to do our inspections, our instruction. We're trying to find out, basically. We're in the process of discovery still, very much so. And that's okay. That's part of the way science is designed to work. It's designed to be exploratory that way. And I think sometimes when you sort of theologize evolution, which many people do, as a matter of fact, this, by the way, is coming from somebody who has their degree in evolution and ecology from UC Davis. So I admit one of my biases is genetics. One of my other biases is I'm not a phyletic gradualist. Intellectually, it does not make any sense. And I'll explain why as the semester goes forward. But UC Davis was, right? So, but the idea is, as we gain in knowledge, then it makes our ideas stronger. It throws into a new light, maybe things we used to think were so, but it's like, wait a minute, it doesn't really work out that way, does it? Because that shouldn't be possible if it was the way I thought it was, right? So it kind of, that sense of constant discovery constantly puts us into places where we're constantly trying to sort of fine tune things, and we're trying to figure things out. And so I always like to do that just to let students know that we're still working on this one, right? Shane, did you have a question?
I was wondering what the big word you would use was, but
For what? Oh, the phyletic gradualism. So phyletic gradualism is kind of, basically in a nutshell, it's kind of your classic Darwinian model of evolution, where organisms that we largely studied, which phyletically, that means we study their morphology, that is structurally how they're similar, how they're different, and we kind of create these sort of ancestral relationships between them. That is largely from a genetics point of view, a post-genomics point of view. I kind of am seeing that more and more as an antiquated sort of a way of looking at things, because ultimately you're just looking at structure, but you're not looking at the root cause of that structure. So when you're talking about things changing form, what underlies and underpins that change in form is genes. So it's your genetics that changes as these species are speciating. And so in reality, if a more authentic view of things is to not look at it, you know, like fossils, for instance. I'm not a fan of fossils. I never have been, because I'm a genetics guy, of course, right? Because you're just looking at a bunch of dead stuff from, you know, who knows how long ago. In some cases we do know, in some cases we're still working on that one, right? But I mean, obviously hundreds of millions, if you're talking about Precambrian, you're talking like a really long time ago, right? I mean, so you're looking at stuff that has been gone for a long time. But you're kind of a forensic study. So you're kind of trying to piece it together as best you can. But it's really, really limited because you're kind of going on shards and scraps when what we really want is to have been there so that we can put it under scientific inspection, but we're not. So we're kind of guessing in the dark a little bit, at least in terms of the nuances. And you can see that in their process, you know, and we have to be honest about that, right? Because that's where the objectivity comes in, because if you're not objective about that, then you're not going to grow. Your ideas aren't going to grow stronger and more robust and more accurate because you're going to be too busy, like, you know, fighting off demons. And that's theology, right? That's a different discipline. And that's the reason why I'm up here, is because there's a lot of work to do here scientifically, right? So, but that's, and I'll kind of touch on that again, right? But so that's kind of the idea of kind of understanding a lot of this. We want to make sure that we keep everything in balance. And one thing I am not afraid to do is I'm not afraid to poke the bear, right? So where I see a weakness, I'm going to point out the weakness, you know, and maybe a dyed-in-the-wool, you know, evolutionary theologian of sorts. That's not an actual thing. That's my sort of sarcastic mock thing. And the reason why I say that, by the way, if I've got an edge there, there's a very good reason why. Because I actually remember very clearly, and to this day I remember this, my professors, some of them, like emeritus professors, guys who've been like great work for decades at UC Davis giving talks about Darwin. And I kid you not, they were so religious that literally you could replace the word Darwin for any deity's name and you would swear you were in church. That is absolutely out of bounds. That's what I mean, and that's what I'm thinking when I say, you know, these sort of theological evolutionists, right? Evolution is an idea. It's a scientific piece that we're trying to work on to explain the world around us. If you start waxing eloquent and religious about evolution or Darwin, then you've just taken yourself to church, right? Which, generally speaking, that folks who are atheists are probably bristling at that comment. But that's basically what's happening, right? Because you can't do anything. If you deify Darwin, if you theologize evolution, you can't do anything with it anymore. You know how I know that? Take a look at all the theological fights that have happened in all the, you know, just religious denominations. Ugly battles. Ugly battles. I mean, just the war between the Catholics and the Protestants, right? I mean, bloodshed. Lots of it. Spanish Inquisition. Remember that one? That was all people disagreeing with each other. Why? Because their view or their interpretation theologically was above touching. That is always dangerous territory to get to. So that's the reason why I'm so hot about knocking down the theological evolutionists. Take your religious passions somewhere else, not in science. Because once you do that, then you can't argue objectively the data that's in front of you. And that's not science, that's advocacy. Okay. But that's important because I can tell you right now, you know, a lot of people put a lot of premium on being intellectually honest, right? Being consistent about things like this. This is what this is about. Be intellectually honest. This is a bit of a sermon also, so to speak, right? Quote unquote, pun intended. Right? This is kind of like a little bit of a preach to the evolutionary biologists themselves, because a lot of times, because of their ego, they don't even see how biased they've become. And that's not healthy, right? One thing in science that you never do, never fall in love with your idea. Because the second you do, you're no longer a scientist. You're an advocate. That's a different thing. What? Huh? Yes, yes. As soon as you fall in love with your idea, you become biased. And so you're an advocate. You're no longer a scientist. And that's a different arena. Dawkins, for instance, is a perfect example. Many of you know Dawkins. How many of you guys know Dawkins? Richard Dawkins, kind of the premier. Every generation, they've got sort of their lead sort of atheist spokesman for all things atheism. And Dawkins is largely started off as a zoologist, but he is not a zoologist. He's not a scientist anymore. I think he would probably call himself a scientist, but he's not, because if you read his books, they're all about theological ideas. He's arguing the case for atheism and materialism and rationalism, which are his main things. But it's an advocate, right? He's an advocate for those sorts of things. And he's a perfect example. I'm not saying that's bad. It's just you're moving into a different arena, right? That's just a different place that you're going. And so it's helpful to always maintain that awareness of kind of where you're going. That's also, by the way, very helpful, because I can guarantee you're not going to get a lot of this discussion in college, which is unfortunate, because you probably should. Why? Because even in just general life out there, how about like the 2020 election, right? When the passions are running high and hot and angry on all sides, right? Being intellectually honest and trying to be fair and open-minded was a hard one to do, right? When you realize that the dog in your race, or the dog that you had in the race, was not as great as you thought, or maybe it was better than you thought. I don't know, right? But having that wherewithal to say, listen, you can't just lie to me, right? That's an important thing, but that has to do with your intellectual honesty. That was a little further than I was expecting to take it, but the problem is there's very few times, I think, when somebody's willing to look in the face and actually say it straight out. And to be quite honest with you, it needs to be said more, because it's not really a comfortable place to be. But you want to make sure you're honest with yourself. So basically, then when we're looking at the diversity of life, ultimately, this is like 19th century, right? And so we kind of like these kind of philosophical underpinnings that we've just been talking about in science largely kind of came together slowly over time. Okay, they weren't quite there yet when Darwin was around. There, some of it was there. But a lot of that kind of gelled together over time. And what's happening now is you're kind of reshaping that a little bit, and we'll talk a little bit about that here in just a second. But in the 19th century, what you had was this concept of diversity that was pretty limited. I mean, yeah, maybe you knew what a lion looked like, but probably your average kid, if you ask them now, draw a lion, a picture of a lion. Probably most of them could pull that off, right? I mean, assuming they're artistic enough to get that, beyond stick figures. I'm still there, by the way. It just looked like some dude with a lot of hair, right? With a big man. That's probably what it would look like if I tried to draw it. But I mean, most of them would probably have an idea. But back in the 19th century, probably many folks never saw a lion in real life, even well into adulthood, much less knowing anything about all these little critters that are out there that, we're kind of discovering. So early in the 19th century, like when Darwin actually went on his famous voyage on the Beagle, he was basically what you refer to as a natural historian, which we kind of identify now as a zoologist. Basically, he, among others, John Muir was one, Audubon was another one, right? They would kind of go all over the place. They'd pick their place that they liked. And then they would just collect stuff, right? They would collect a bunch of animals. Usually they would probably collect them and then they'd kill them. And they bring them all back. And then they would kind of study them, right? Because they didn't really have the wherewithal to do that. Which is a little unfortunate. I mean, we don't really kill them now, but that's unfortunate what they did. How many of you guys are familiar with Audubon's work? The bird guy, although of the original, he's like the one who kind of basically painted pictures of birds of all over the place, but they all look weird. Look up Audubon. All those birds were like that. And the reason is and no natural bird looks like that, right? I mean, you kind of see them all like, they're kind of like, but these are guys are all like splayed out. That's because they were dead. So he was painting a picture of a dead bird that he just kind of like threw out there or kind of moved around so he could see detail. But that's the reason why they look all like tormented, is because they're all dead. But that's what he did, right? He cataloged what he saw. And this is the first time we actually got a sophisticated catalog of all these things and of all these organisms. And that's what Darwin did, right? So Darwin brought all these things back. He had a lab full of stuff and he was just trying to figure out, what's the relationship between all these? And that's kind of where the whole evolution idea came from, as he was trying to make sense of all the stuff that he collected. And that's one very, very important idea, is the sense of diversity, the fact that living things in the world are not all the same. And there's a lot of differences out there. And so the first thing that we did when we saw this diversity is probably something that you would automatically do just naturally, right? You got a bunch of stuff you've never seen it before. What do you start doing? Yeah, you start making sense of it, right? So you start organizing it. And that's exactly what we did. And basically Linnaeus essentially invented the discipline of taxonomy. And this is basically the classification of species by their variable morphology. It goes something like this. You got a bunch of stuff, right? Okay, all you cat-like animals, you go to that corner. Dog-like animals, you guys go to that corner. Bear-like animals, you guys go to that corner. And I'll kind of start sorting through you here in just a second. And then you kind of start sorting through them a little bit, right? So that's what taxonomy is. And so you kind of start to create these categories of all these organisms based on their morphology, the similarity, the fact that they look similar to each other, right? And so that's kind of where we get our classic taxonomic system. And then, of course, what we did was this is a growing system, by the way. So Linnaeus began this, in the 19th century. It was early. It was early on. I mean, he started organizing what he knew. We just add to it, as we started discovering new species and things like that. And so we just kind of keep adding to it. And so taxonomy is really a growing discipline, right? And so there's a lot to be made about taxonomy. It's not intended, taxonomy was never originally intended to do what Darwin does in evolution, which was to pin down the relationships between ancestral lines, right? So Darwin was trying to draw inferences of relatedness between two species and their nearest ancestor. That's what Darwin was trying to do. But in taxonomy, Linnaeus was just categorizing stuff. It's like he wasn't really looking at relationships. He was just saying, like, I don't know how you guys related. You just kind of look the same. So you kind of look like bears. So you're in the bear group, right? So that's it's much, much more just kind of like a categorical sort of a thing. And so what that gives us then is a more sophisticated taxonomy. Now, the thing with taxonomy is that you would imagine then that as you discover more species, it's going to change your classification because you're going to learn more about these organisms and you're going to see stuff you never accounted for. So you have to create new categories. You're going to have to sort of shuffle some things around as you learn new things. And so what we have here is basically what we currently have in terms of our taxonomy. And the taxonomy, basically the way this works is essentially it's like a biological 20 questions, right? How many of you guys have played 20 questions? Yeah, right. It's pretty simple. You got 20 chances to guess who I'm thinking about, right? And the questions that you ask are what types of questions? Yeah, they're binary, right? So there's two possibilities. So oftentimes it'll start off with like, are you a male or female, right? And you'd be like, okay, neither, right? Which is not binary, but I mean, it's, that actually probably makes it a whole lot easier, actually, because you just pretty much eliminated a huge chunk of the population and narrowed down a very small subset. But typically, historically speaking, you'd be like, okay, you're male or female, right? So, okay, I'm male, right? So what's the next question you like to ask? That's a good one. Yep, that's a good one, right? It's helpful, right? Are you alive still or not? So it'd be like, okay, I'm alive. Usually you guys will get to another one, a common one. Are you famous? Right? Because otherwise, if you're not famous, then how in the world am I supposed to know who you are? Okay, maybe that's not a good one. Maybe the better question is like, are you an actor? No, I'm not. Are you a singer? No, see? That's unfortunately, that's a little interpretive. So my sense of popularity could be different than yours. Right? So so you basically after 20, hopefully you get down to a specific person. Got it, right? Well, this is a biological 20 questions. What's the first question? Yeah. Generally, well, this is recorded. So you can also see it as well if you don't catch it all. But generally speaking, as a general rule of thumb, if I'm writing it down, you should probably know it.
Oh, so this is kind of like a screen recording that's gonna go to our notes.
Yeah, this is actually what gets loaded in the recorded lectures.
Okay.
So, yeah. Yeah, so it's recorded just to make it easier on you guys, but like any kind of notes that you write to help clarify things in your own mind, that's all stuff that, you know, you can focus on. So the question is, are you prokaryote or eukaryote? Well, that's a good question. I don't know. What does that mean? So as it turns out, by the way, there's only really two types of cells in the world: prokaryotic cells and eukaryotic cells. Prokaryotic cells are defined by very, very simple single-celled organisms. And they don't have a lot of organization to them. So they're just kind of like one, like bacteria is a good example, right? They're just basically one single cell. They don't have any compartmentalization in them. They don't have, they don't have a nucleus and have organelles. They don't have any of those sorts of things associated with them. They're very, very simple. And they're very basic. A eukaryote, however, is much more complicated. They have compartments in them, organelles, where you have division of labor. So you do different types of tasks in these organelles. And so they're much more complicated. Where prokaryotes are very, very tiny. You need to use like the 100x objective. That's what you're usually studying in microbiology. Eukaryotes are fairly large. And as it turns out, you basically fall into one or the other. Now, as we sort of develop this, what we will notice is this defines, once you answer this defines the domain level. And in the domain level, there are two prokaryotes actually that we have. There is archaea and bacteria. So bacteria, like what we associate with, like E. coli, salmonella, staph, those sorts of things. Those are all prokaryotes in bacteria. Archaea, we only find at things like deep sea thermal vents, right? So they're extremophiles. They're in really harsh environments that we don't actually think things should be able to live, but they do. And so that's where we find archaea. This is actually a fairly recent discovery, archaea is. So when I was in college, we didn't have domains. We had 5 kingdoms. Monera was one of them. But this didn't exist, this level. So we learned something and we had to change the system. Okay. Now, the third domain is eukarya. And that contains us. So we are eukaryotes. So we have organelles in us, and compartmentalization. We're more complicated, we're more structured. And so there's a lot more that we can do. Now, ultimately, as you go through the 20 questions, you will see that you have a different sort of what's called a taxonomic level, depending on the question that you're asking at that point. So there are more. Right now, that's our taxonomic list. Each one of these is defined by a question that you're asking. Here's just a couple. I'm just going to tease them out. I'm not going to tell you which one, what level they're at, but just to give you an idea of some of the things that you're doing. One of the questions that you would ask, say, for instance, is kind of what's your biological plan? Do you have a distinct biological plan? Typically, that will break you up into kingdoms, right? So from domain, you break up into kingdoms. So under eukarya, which is more interesting, you've got 4 kingdoms because you have protista, which is typically composed of single-celled organisms that have a unicellular lifestyle. Okay, some of these protists are animal-like, some of them are plant-like. Okay, but they have a unicellular way of living. Now, you can see you've got a little kelp there in the picture. So you're probably thinking, wait a minute, but you said these guys were single cell. What about the kelp? I can see that, right? Well, kelp is actually a colony of single-celled protists, algae, actually, in particular, brown algae. And so colonies of cells doesn't count as the organism, right? Colonialism is something that happened a little later, but just because you're a single cell and you hang out with people doesn't necessarily mean that you become one entity. Just because you guys are 1111 students doesn't mean that you guys in this classroom have all basically melted into one entity, like the Borg for you Star Trek fans, right? You don't have a hive mind yet. You are all very much individuals, right? But you're kind of colonizing. That's what protists do. Think pond scum. So you see a stagnant pond that's green and slimy. That's lovely. That's like biological gold. There's all kinds of protists swimming around in there. Giardia. How many of you guys have danced with Giardia? Yeah, go up to the mountains, drink the water. Get Giardia. What? That's a protist. That's the reason why you have to treat the water, is because it happens to be, in this case, a parasitic protist. So that'll wreak havoc, especially if you like to run to the facilities on an every other minute basis. And then end up in the emergency room to get fluids because you'll probably dehydrate and die if they don't stop it. So fungi, those have a completely different way of living, right? They kind of got a mushroomy thing going on. Fungi, yep. So I know fungi, but yes, they got a completely different way of living, right? So mushrooms, molds, yeast is in kingdom fungi. And so they have a completely different way of living that's different than plants who are in the kingdom plantae. So they've got a very distinct way of living and doing things. And then of course, animals, which we probably know the most about. Why? Because this is where we are, right? So we kind of understand that one. And so we have a very different way of living than plants, very different way than mushrooms, very different way than protists. Okay. So at the kingdom level, you're kind of asking the question, do you have a different style or a different plan for living? And we break you up into kingdoms. Now, as you kind of start going on, let's just to pick on the animals here for a second, just to give you an idea of how we start to parse these guys out. You know, at one point, at one level, we'll ask the question in animals, not necessarily in order, but we'll ask an important question. We'll say, okay, fine, you're an animal. That's great. But do you have a backbone? Yes or no? The answer is yes, then you become a vertebrate. And then we start to sort the vertebrates out. If you say no, then you become an invertebrate. Insects, right? Bugs. By the way, the invertebrates far outnumber us, like by a lot. What? Do you mean that live in the water? Yes. Doesn't matter where they live. If they live in the water or if they live on terrestrial habitats, doesn't matter. If they don't have a backbone, they're invertebrates. Jellyfish would be an invertebrate. Yeah.
Really?
Mm-hmm. Because it has no backbone. An insect is an invertebrate because it doesn't have a backbone.
No, I meant the jellyfish.
It's not, no, it's not, but that's a different level, right? Because then you're starting to separate your invertebrates out and saying like, okay, so what are the bug-like invertebrates, right? So spiders are also invertebrates, but they're not insecta, they're arachna, right? They're arachnids. So you kind of further separate out your invertebrates further on. Yeah. So there's still sorting out to do. It's just kind of, you're playing with the invertebrates at this point. That makes sense? So the same things with the vertebrates, right? Then you can start to ask yourselves other questions like, for instance, are you a mammal? Well, what does that mean? Okay, first of all, do you give live birth and do you have mammary glands? Yes or no? It's either yes or it's no. If it's yes, then you're in Mammalia, right? That's another taxonomic division. If it's no, then you're over here. Maybe you're an egg layer, okay? Like turtles, for instance. And so then you kind of further parse them out. So you can say like, well, okay, wait a minute. So are you, say you're a mammal, but are you, do you eat producers, right? Are you herbivorous or carnivorous? So then you can separate them into carnivora or not. So carnivore, meat eaters basically. And you can kind of keep separating them out until eventually you get all the way down to something like this. Where this is your genus and this is your species. This is what's called the specific epithet. Anybody know what that is? Yeah. Yeah, you guys probably, some of you folks had this at home, right? Now let's compare this to that one. Who's that? Yeah, it's a wolf, right? So still a canid, basically a dog-like creature, but not the same species, right? Okay, so this is just to give you other folks some love here. What's that? Yeah, that's your cat. That's for the cat lovers, right? So don't want to throw the cats out there. But that's Felix domesticus, that's your house cat, right? But this is an important thing, right? Because whenever you get your species name, right, Canis familiaris, or what's us? What's our name? Homo sapiens, right? So genus is Homo, sapiens is species. So by the time we get down to genus species, we have an entire laundry list of names, right? So we have a domain name, we have a kingdom name, we have a phylum name, a class, order, family, genus, and so forth. So there's an entire laundry list of names. We have a very long name, right? We just shorten it to genus and species, which is sufficient specificity to say that in the species, there's no other species like it. Okay, so they're individual and specific. But are they? So here's one of the pushbacks, the soft spots, I will, not necessarily soft spot like a bad thing, but just like things to consider that I think are oversimplifications when authors and textbooks try to make things like evolutionary studies and things like that too simple, because it's not simple. Here's a good example. Can these two guys hybridize? Yeah, we have the wolf hybrids out there, don't we? So they can hybridize. Generally speaking, when you're looking at species, and like if you're doing an evolutionary study and you're looking at what you look at when you look at evolutionary studies, that is speciation, right? So the change in organisms over time. Then one of the things you have to do at the heart of your study is you have to always be aware and cognizant of how you are defining species, right? So what do you mean by species? And the part of the reason is because in evolution, there's actually probably like about a half dozen different definitions of a species. None of them are wrong, right? They're just alternative definitions of the species. But the problem is, depending on which definition you use, it will shade your results. Generally speaking, for instance, when you're using just what's called the biological definition of species, that is to say the two species should not be able to hybridize, right? That's reproductive isolation. Here's a good example. Let me do something a little easier for you guys. We are Homo sapiens, yes? We have Canis familiaris, yes? Are these two clearly separate species?
Yes.
Right, so Canis familiaris and Homo sapiens will not hybridize. Clearly, they are two very isolated, very separate species. The relationship there is very distinct and clear, right? Something like that we feel very comfortable with. However, when you're looking at speciation within like a generally related taxa or something like this, then the question is, how do you define species? Because how you define species is going to shade the results of your evolutionary discussion. Does that make sense? And this is very difficult because a lot of times as we learn more about these organisms, we change our taxonomy system. All the time, actually. Matter of fact, I gave you an example of it, right? Domain didn't exist when I was in college. We learned a little bit more about organisms and we had to create a new taxonomic level. So it's a new taxonomy. This happens all the time, actually, in ornithology. Here's a good example. When I was at UC Davis and I took my ornithology class, one of my favorite birds, which we have around here, was the rufous-sided towhee. Stunning bird, all black on the back, kind of looks like a little tuxedo sort of a thing, white breast, kind of a nice, generous, rusty little spot right here on the side. All black head, blazing red eyes. If you look at them, really cool. There's two variants to that. The rufous-sided towhee, which was one species when I was at UC Davis, has since been split into two species. From about probably the Central Flyway west, we have the spotted towhee. So in Colorado, you guys probably have seen this bird actually in your backyards. If not, look for them. They're actually quite attractive. They're about maybe yay big. A lot of ground hoppers. They like to hop around on the ground, so just toss some seed out there, something like that. You'll probably find them eventually. But they have white spots on their feathers. From about the Central Flyway east, they don't have white spots. So the towhee has been split into the spotted towhee, which we have here in Colorado, and the eastern towhee is what we have from the Central Flyway east. So here's the thing. There's two species now. Did the towhee change at all? Evolutionarily, did it change? They still had the same spots in some areas. When I was in college, when it was just the rufous-sided towhee, and some of the areas they didn't have the spots. So the look of the spots versus no spots, that's not new. That was still there when they were rufous-sided towhees, one species. Now they're two species. Did they evolve? No. Yeah, no, they didn't evolve. What we did was we split them, right? And that happens all the time in taxonomy. We'll learn something about these organisms. We realize, wait a minute, these aren't really two different species. They're actually more alike than not. So we're going to lump them together. And then in some cases, we're like, you know what? These two kind of have two variants in different parts of the country. They're actually two different species. So we're going to call them two different species. And so we split them. But the birds themselves did nothing evolutionarily. It was us. We just organized them differently, right? And that's just based on how we define species and the information that we learned. And that's always a trick, right? Because one thing you could argue is like, first of all, the eastern and the spotted towhee are not different species. Why? Because if you put the two together, they're going to have babies, just like any other, right? So they're not two different species. So at what point do we call it actually evolution? Does it get so far distant from each other that we call it actually, okay, they've technically evolved into different species versus they're just adapting to their environment. They're a little different, yeah, but they're adapting. Here's a good example. Scandinavia, where the human population is extremely fair-skinned and light-haired, like outlier-like. Like if you take a look at the human population, they are like rare. They've got all the rare traits. And yet, that's a preponderant look in Scandinavia. So the question is, at what point do you call that an adapting population versus a new species? Exactly. Nobody knows really how to draw that line. Like, when does it stop being adaptation of a single species or a group of related species and actually become something identifiably different that we can say, aha, there it is, it turned into something else. That's always a trick. That's always a difficult sort of a thing.
Oh, no, I thought you were looking for.
Oh, sorry, go ahead. Yeah. So basically then that's that's the reason why I say that is because that shades all of your discussions, and it's helpful, right? Because it's interesting and makes it more fascinating and it makes it more real. It's not as simple as oftentimes we give you, okay? It does, but that's the thing. That's the reason why it's like when I look at them, like, okay, how much time do we have? Because if you want me to explain it, we're going to carve out time to do it right. It's better not to do it at all than to do it kind of half-assed, because it can be misleading. It can be very misleading and very deceptive. And I think a lot of people feel like they got lied to or duped because they didn't actually get the full, fair, deep treatment so they can understand fully and appreciate fully what we're going through and what we're trying to get accomplished. Because honestly, especially in this particular one, because there's a lot of evolutionary theologians out there that want to kind of theologize evolution, a lot of times when they get a little too simple, it actually becomes kind of like you're trying to sell them a bill of goods. It's like, oh, well, I'm not going to tell you how it all works. Just trust me, it's science fact. whoa, that sounds pretty arrogant. Last time I checked, we still do experiments, right? Last time I checked, we're still struggling with the nuances of how to define species and how to record how species are changing and adapting over time and what that means, right? That's still a process, and that's a healthy process, right? Because we're still trying to figure it out. Because to be quite honest with you, we're still trying to figure it out now. In the 19th century, they didn't have anything to go on. It was all just conjecture and guesswork. So we now are able, for the first time really as a generation, able to do these experiments and to try to actually put data to it all, okay? Which is the reason why it's so important now to really kind of like, you know, this is going to get better, but we gotta be willing to question some of our underlying assumptions so that we can shore them up, or if they need modifying, we can modify them and say like, well, it actually is probably more like this because this data is better explained with this model. Good.
What about to distinguish the features between like archaea and bacteria?
Between archaea and bacteria. So bacteria has, in this particular case, these two guys are separated because in the bacteria, they kind of have a very kind of what I say a classic sort of a prokaryotic presentation, right? So a lot of their mechanisms that they do, so like their membrane construction, the way they do metabolism, the way they do things like reproducing DNA, making proteins and things like that, there's a very prokaryotic flair. As a matter of fact, you're gonna learn that. Because in every chapter, when we get through these biological mechanisms, the first thing we do is ask ourselves, how do the prokaryotes do it? Because that was our first stopping point. We studied bacteria first before we studied anything else. And then what we did is we said, okay, now that we understand how it happens in bacteria, how does it happen in eukaryotes? And a lot of times, a lot of those themes were similar, but there was a lot of extra stuff. Okay. In archaea, it's a little bit different because archaea actually shares some similarities in their mechanisms with bacteria, and some of them with eukaryotes. So it has some similarities between both. So it's kind of like somewhere in between. It's like the overlap region of a Venn diagram between the two. So this is basically taxonomy. It's a living, breathing thing. The more we learn, the more we sort of massage it, change it a little bit so we can sort of more accurately describe things. And that's kind of what we're looking at. And then a lot of times how we learn new information about sort of things, relationships and stuff like that in particular, is a lot of times from these evolutionary studies that we do. And we realize, wait a minute, these shouldn't be doing this if they're related this way. So if this, you know, if they're like, if I'm getting this data, then that means these relationships should be a little bit different, right? So that's kind of how we do a lot of this work and how we kind of build this a little bit. So it's changing over time. But this is your basic rundown, right? And so one of the things you want to be able to do is do kind of a, you want to be able to know this list in order. Getting more specific as you go down. And there's an easy memory trigger to be able to remember this list in order. Go something like this. So a little arresting, even if you're not a cat person. I mean, most people still like kittens, right? And so that's a little arresting, but you know, drunk kittens playing chase on freeways get smashed. So you have domain, kingdom, phylum, class, order, family, genus, and species. Drunk kittens playing chase on freeways get smashed. Okay, so that's how you remember it, getting more specific as you get closer to being smashed. The third one is playing, so that's phylum, P-H-Y-L-U-M. There's one L in there. So you're getting more and more specific as you go closer toward smashed, okay? Getting more specific. So like there's a lot of organisms in phylum. There's fewer in class. There's fewer in order. There's fewer in family and so forth. There's only one species in species. Or ideally speaking, right? And the other thing also that I always like to just give you a feel for it is usually when you're looking at evolutionary relationships, normally what you're looking at is you're kind of inspecting change, usually within a subset of this taxonomic scale, right? So That's a good example of like when you're looking down here, when you're trying to parse out the difference between whether or not familiaris and lupus is actually a separate species, and you're trying to do maybe some work to try to tease that out, try to understand, how those species are working and interacting, that sort of stuff. Generally, I always kind of use family as a marker, and a lot of folks will use family as a marker. A lot of evolutionary biologists will use that because at family, this is about the range. Like if you were to look at an animal you've never seen it before, and you look at it and you're like, I don't know what that is, but it's kind of dog-like, right? And you're not quite sure what it is, but kind of it's something of a doggish nature. Usually when you're thinking of that level, you're thinking at the family level. So all the dog-like organisms, Canidae, are in that family taxonomy, and then they kind of parse them out in genus, separate them out, and then go to the species. So that's usually a lot of times where you go. And so a lot of evolutionary biology, especially when it's tied to ecology, is basically from family on down. Okay. And we'll talk more about that. And some evolution, they'll be working up here as well. But the problem with this one is that stuff is a little harder to wrap your head around, just because there's so many things in it, right? So if you want to study a phylum, it's like there's just a ton of stuff in the phylum to sort through. So it kind of gets a little more manageable as you go through into the smaller levels. So the other thing I also want to do is kind of help to kind of define then, and this kind of, I'm still kind of a little bit philosophical. I'm going to get more concrete here as we kind of go on. But one of the things I always want to sort of focus on is, as we sort of talked about taxonomy, is to just kind of underpin, when we take a look at life, what we notice is that living organisms are not easy. There's nothing simple about a living organism, even the simplest ones that we have, right? Like prokaryotes, for instance. Just looking at, say, for instance, DNA replication in a prokaryote is an enormous undertaking, and it's very, very difficult. And even in real time, and I want to mention this as well, just to kind of shade this and make sure we got our perspective straight. And this is like me poking the bear. Like I said, I'm not afraid to poke the bear. I'm going to call it like it is. And you're free to, anybody can disagree with me. A lot of my colleagues do, but it makes for good spirited conversation. But I've got reasons for saying what I say, and I've got reasons for being where I am. So it's not arbitrary. But one of the things, especially now in genetics, especially, it's like when you're wired for genetics and like biochemistry and cellular biology, one of the things that's screamingly loud at you when you're studying on that level is that there is a lot of complexity in life. I mean, it is an amazingly complicated world, and like a jaw-dropping complicated world. There's a lot going on, and it seems like there's no way in the world why any of this should make sense, and yet it all fits together perfectly. It's really an amazing place to be, but one of the screaming messages is this is hard stuff. To understand what's going on here is difficult, right? So when we're looking at, say, for instance, a cell doing its thing, and we're trying to understand some biological mechanism, it's difficult for us to understand it even in real time while it's doing it in front of us and we're measuring it and we're analyzing it. It's very difficult. Now we've gotten good at that. How do we know that? Because all of that real-time research has borne fruit, hasn't it? Anybody enjoy modern medicine lately? Yeah, all of the medications, basically all of medicine as we know it, has been born on the shoulders of research. So basic scientists studying how things work in real time is what develops and evolves, intellectually evolves, not biologically evolves, into a therapeutic that we can then use to help us with our diseases. The COVID vaccination development is a perfect example. That's where our knowledge of coronaviruses and virology hit hyperspeed. We pulled it together as a group and as a country, and we were able to basically develop a vaccine in historic time. I don't know if you guys were paying attention, but what happened there to develop a vaccine for the coronavirus has never been done in history. Coronaviruses, we don't have vaccines against coronaviruses. And we were able to do it within months. That is because we pooled everything we understood we kind of cracked this open and we figured out what was happening in real time, and we're able to develop that into technology that benefits us. But it was hard, because we would not have done that in a few months if we hadn't had so much work in virology that had gone on decades before. So all those virologists that nobody's going to know of, they were the ones who laid the groundwork to save the world. Right.
Don't know that there's Ebola corona.
Yeah, there's actually quite a few. Yeah, Ebola is one of them. Yeah, Ebola is a coronavirus. There are other coronaviruses that are much nastier. Not that that makes us any of us feel better, but yeah, it's not like new. The coronaviruses are all around us. So most respiratory infections actually are coronaviruses of one stripe or another. So they're not new. This is the first time we'd actually been able to develop a vaccine against it. So that's an amazing thing, right? That tells us that this is not complicated, because it took decades of footwork ahead of time in order to put all that together and actually make that happen. We didn't just create this and discover this like in months. That's not the way it happened. We took everything we learned from decades before and pulled it all together and just turned it into high speed. It was kind of like it was the biological version of what happened in the 60s with the space program. Kennedy announces it. Less than 10 years later, we're on the moon. Right? That was basically what happened in 2020. The researchers had an idea of how to do it, just like the space people had rocket knowledge and all that sort of stuff. And they just kicked it into high gear and they pulled it all together, and we had a virus vaccine within a year. Okay, that is preposterous, and people should make note of that in history, because that was a big deal. Okay. Unfortunately, the reason why nobody knows that is because there was a rather unpleasant election going on at the same time, and that's all that anybody ever remembers, which is unfortunate. Maybe in time, people will grow up and will remember what's important. Yes, that's me poking a bear. Not quite sure which bear exactly, but there's a bear somewhere that I just poked. But okay, so it's really, really difficult to know what's going on even when you're studying in real time. Now, here's the thing, right? And this is kind of like one of those things that kind of is one of the soft spots that I would say when you're kind of working just with evolutionary studies and things like that, is the further back you go in time, the harder that gets, right? So we have a hard enough time in real time understanding how things work specifically. As we start to go back hundreds of millions of years, or a good example is like the like studying cellular evolution, right? The first cells that came along, I mean, happening some 4.5 billion years ago or so. I mean, that's just so far back in the past that it's difficult to really know for sure exactly how it all happened. Right. So, and that's just because there's nobody there. There's nobody there, nobody to record stuff the way we need to record it for science to be able to understand exactly how it happened. I mean, all we have are sort of these leftover tidbits, these little nibbles of things that that we kind of have to sort of hypothesize around, right? And so the further back we go, the more and more impossible it gets to test it. Remember, that was the second requirement of science. You have to be objective. Number one, it has to be testable. And that's kind of the soft spot of looking back retrospectively, is because when you're looking back and trying to recreate an event that happened that far back ago, it's almost impossible to know for sure how that worked. And not only that, but it's certainly not testable, at least not in a discrete sense, right? So you can't actually say, aha, this is we're a test that's going to tell us that this happened this way or this way, right? So what you end up having is a lot of guesses. Well, that's a little too cavalier. What you have is a robust hypothesizing effort that utilizes your knowledge of how things are to try to piece together how things could have come about. And there's something, and what you'll see the further back you go is oftentimes competing ideas, right? So you'll see multiple versions of the story is what you'll see, because it's not possible to pin down any one and say, aha, this is exactly the way it happened. Because ultimately we weren't there, we're not quite sure, right? We have some ideas that it might have happened this way, but the one thing we use in science when we have to sort of reach back like that is something that's called explanatory power. And explanatory power is the capacity of an idea to explain current phenomena. So for instance, if I were to look back 4.5 billion years ago, and I were to create a model, right, or hypothesis, or whatever you want, that says, well, I think it kind of came together this way. And the question would be like, okay, that's great, but how do you know, right? Because if you're just sort of making up a story, how is that any different than just like mythologizing, right? Anybody can make up a story. So how do you know yours is right if you can't test it, right? Right. So what you have is what's called explanatory power. So you basically say, it's like, well, if it did happen this way, then it would explain why we see this, and this today. Right? The more things that your idea can explain, the more explanatory power it has. That's the idea. And generally speaking, when an idea has more explanatory power, it makes it more robust. Now, does that necessarily mean that that's it? No. I can tell you right now, the cellular evolution, like 4.5 billion years ago, we'll talk about more about that when we get to the cell chapter. There's several competing versions of that idea. Nobody knows actually which one is right for sure. And honestly, nobody really knows if there's something completely out there that we haven't considered yet that is actually the way it went, and that we're actually wrong on all fronts. But that's kind of the way it works. So it's a constant process. That's what I want to say. And so it kind of requires that sort of intellectual honesty, right? So you have to be willing to admit, okay, so this is where my theory is weak. These are the assumptions that it rests on. I'm not quite sure if those assumptions are robust enough. I think they are, but I have to also be honest. I have to be objective. It may be some of those assumptions aren't robust enough, right, to hold up my theory. And you kind of have to be honest with yourself, okay? And that's important. Here's the reason, a really good application. One of the reasons why you have to be objective and honest with yourself. Because sometimes in science, and I love these moments, you discover something that turns your head upside down. And that's a good thing for science because we get so egomaniacal. Researchers are typically very egomaniacs, and they're also socially dysfunctional. So they got big egos. They used to be the smartest kid in the class. They don't like to be wrong, which sets them up to fall in love with their idea. And we all know what happens once you do that. But here's the thing. A recent discovery is a perfect example of maintaining your objectivity. Because if you do, awesome things can happen, right? I mean, the truth is going to be more fascinating than your idea, right? That's where you want to go ultimately. You want to know how it actually works, not how you think it works. And you want the two to be the same, ideally, right? But a recent discovery that really kind of turned things upside down in evolutionary biology, which I thought was fascinating, is the discovery of T-Rex fossils. Now, how long did T-Rex live? How long ago?
While, like 7 years.
No, like how long ago was the last T-Rex on the planet? It does go that, but like the youngest one, the most recently possible. Think Chicxulub. You're probably looking at least at the youngest, right? You're probably looking at 65 million years, right? So they have a T-Rex fossil, and actually Mary Schweitzer discovered this in North Carolina State. Awesome, she's doing some fascinating work. I love this story because it just turns us all upside down and forces us out of our kind of our delusion that we've got it all right, our status quo. But what she found was red blood cells in T-Rex fossils. So for those of you who know what that means, does anybody know what a T-Rex fossil is made of? No.
Oh, rock.
It's rock, right? The mineralization process, which is the replacement of the minerals of the rock for the minerals of the bone, is a conversion process that should have happened. I mean, at most, within certainly 1 million years, right? So every single fossil you see in a fossil bed should be rock. Just like the body casts of Pompeii are not real people. It's the pyroclastic ash that basically has sort of replaced the body, right? It's hollow inside, but yeah, but the pyroclastic flow, because of all the fluids that basically, when they burned, basically mixed with the pyroclastic ash, making cement. So it kind of created like a little thin cement shell with the body fluids, basically. It's pretty sick, but with the body fluids, right? But it's empty on the inside because that would have been like their bones. They never have bones in there, perhaps maybe inside there, but it's everything else is largely since gone. So it was replaced, right, with rock. Just like with bones, when you fossilize, the minerals of the bone get leached out and replaced with the rock minerals. So when you look at a fossil bed, when you're digging out a fossil, you're not digging out bone. You're digging out rocks. Not only that, but soft tissue is not stable across long periods of time. I mean, a red blood cell, they also found collagen, which is a protein associated with your connective tissue. So you have protein and blood cells inside the T-Rex fossil. So that, by everything we understood before this discovery, should not be possible. That fossil should have fossilized. It should have mineralized millions, tens of millions of years before. The protein should have degraded long ago. So the fact that there's protein in there and there's red blood cells in there kind of turns the entire notion upside down. This is a jaw-dropping discovery. Because now all of a sudden you're looking at these fossils, and the reason why nobody ever picked this up is because everybody just assumed it was like, oh, well, these are all rocks, so why would you demineralize them? Because what happens is when you demineralize them, you'll lose the fossil, right? So you have a process that just sort of like dissolves away the rock, and so of course, if you're a paleontologist, you lose your bone. So why would you do that, right? You just basically dissolve your bone. Of course, you're thinking mineralization has finished millions of years ago. So you just basically be mineralizing your pretty little fossil into nothing. Why would you do that? Well, Mary did that for some reason. I don't quite catch the reason why she did that originally, but she did that. And then she makes this discovery, which is just jaw-dropping. It's absolutely flabbergasting because soft tissue, red blood cells, collagen should not last 65 million years. At the very least, this backs everybody up, and they're like, okay, we got questions. Oh, lots of questions, right?
We didn't.
Yeah, that's what they were finding.
Yeah, it's still soft tissue. It's part of the bone.
Yes, because soft tissue does not mineralize. It decays. It's supposed to decay. But clearly, there's something going on that's challenging our previous status quo knowledge of how mineralization worked and how this whole process worked. So it kind of threw a lot of questions in our mind, which I think is fascinating, right? Because it forces us to ask these questions anew, and we're able to get new knowledge. Who's saying? You had your hand up.
So the mineralization process is what preserved the collagen.
Well, the mineralization process doesn't fossilize soft tissue because soft tissue doesn't fossilize. It just decays, right? So that's the reason why we don't see like cells in sediment and things of that nature, is because for the most part, it doesn't preserve. It just decays because there's a lot of stuff out there that destroys it, which would be awesome because everybody would love to see that, because that would really help, especially for like the 400 billion year ago ideas that are competing with each other about how cells evolved. I mean, if we had a fossil, like a primordial fossil cell in sediment, that would be it. That would be what we want to find. But we don't see stuff out there. We see organisms, by and large, mostly formed organisms in the sediment. That's mostly what we see. But this has a lot of questions to it, right? I mean, think about this. Here we thought these things were rock, but they're not. So these things are actually in bone that hasn't decayed. And so now all of a sudden you're looking at your fossil bed and you're thinking, wait a minute, are you guys all rock? Or are you also, do you have tissue in you? Do you have collagen in you? Do you have red blood cells in you? Right? Not only that, but if we have collagen that lasted this long, DNA is actually more stable than collagen. It's more stable than protein. So is it possible that we'll be able to stumble on a well-preserved DNA sample in one of these fossils? I'm serious, Jurassic Park is no longer a joke anymore. We already have cloning technology. If we were able to like sequence the entire T-Rex genome, we could essentially clone and bring to life T-Rex for the first time in 65 million years.
There's a company in Europe right now called trying to bring out the
Yeah, mammoth is easy because we got entire preserved specimens. Yeah, so that's a they're getting closer. I think, yeah, they're getting close to that one, right? But this is cool stuff, right? Because like, first of all, this is a perfect example. Maintain your objectivity. Because when you get thrown that curveball, which every single time you think you've got it right, watch out, because you are about ready to fall hard. And scientists fall for it every single time. I mean, we are not smart in science. We feel like we get a bunch of answers, we're doing pretty good. Then we're like, oh, we got it. This is it. And then something like this happens, you get some crazy curveball thrown at you, and it just turns everything upside down, and you're kind of like starting back at zero. And the ones that emerge to kind of build the new, more accurate reality are the ones typically who remain and maintain their objectivity, because the other ones die defending their idea and nobody remembers them. It can be brutal, right? That's the reason why I say maintain your objectivity and ask questions, ask hard questions, right? Because I mean, as soon as I saw that, I'm still asking questions. Like, it still blows me away. It's like, this is still not possible. It's still not possible, right? Because even if you say that there's some sort of a high preservation, the question is, how many bones are we talking about here? Are we just talking about an outlier in this one deposit? Okay, I could go for that. Or are we talking about major bone beds across the world where we're seeing these deposits of protein? That's a different ball game there. Then all of a sudden you're thinking, well, wait a minute, we didn't just get some sort of an isolated weird hiccup in mineralization. We're talking about more of like a kind of a categoric misread on mineralization across the board at a very deep fundamental level, which changes the game. I mean, and that's exciting, because then all of a sudden, I mean, are you kidding me? If we could see a real T-Rex walking down the street in my lifetime, that would be awesome. It would be Jurassic Park. I mean, and not only that, but you know the scene in Jurassic Park, my favorite scene, by the way. There's a lot of good ones. A lot of people love T-Rex scene. That's a good one too. But my favorite, my nerd favorite, right, is when the paleontologist, whatever his name is, I forget his name, but when he is watching some dinosaurs, and it's like when they first see the dinosaurs, and it's like and he looks at them, he's all like, oh my gosh, they do it this way. We had it all wrong. The reason why I love that is because those moments when science, which is very difficult to get them to because they're so conceited in science, because we think we have all the answers, and we have a lot, no doubt, but not all. But when you have that moment where you just step back and you stumble back because you're just like jaw drop, and you're like, oh my gosh, it's like we weren't even close. It's like the way we thought it happened isn't the way it happened at all. Here it is. Here's how that's happening. Those moments are so exciting because it's like, for the first time, it's like you're seeing the way it actually happened. That's about it. I mean, that's like just an amazing moment. That's that scene right there, that scene gave me chills, because it's like, oh my gosh, for the first time, it's like we see how it actually worked. It was like amazing. I know the rest of us like T-Rex, but T-Rex is cool. But that was my moment, right? Because I can tell you, we have those moments actually periodically through science. Science goes through cycles. We start off dumb. We learn a lot through scientific experimentation. We get a little delusional and conceited with ourselves about how much we know. And then we discover something that just completely scrambles our brain. And then we start back again where we're dumb again. And we're having to sort of reimagine everything, and we get all conceited with ourselves again. And then we make some sort of a discovery, and we scrambles our brains. And then we have to start all over again. And this is one of those brain scrambles. And usually when we scramble our brains like that, and it forces us to step back and sort of reimagine everything we thought, those are healthy moments, because we're growing as a scientific discipline, and we're understanding the world a little bit more accurately. By the way, if we hadn't scrambled our brains and tried to reimagine the world, we would not have made any of the medical discoveries that we made that we all enjoy. Okay.
I was interested in one thing about so they just have never destroyed a fossil before. So, I mean, just go around destroying fossils. I mean, wouldn't that like bring out a curiosity to go start making stuff?
It would. It would. The problem is, I mean, fossils are almost not necessary because we have technology today where we can basically recreate the fossil, at least from the fossil structure itself.
Like as it's like the importance of finding it and like, oh, it's so cool.
Yeah, I mean, once we dig it out and we've actually got this fossil here, I mean, we can recreate that fossil pretty accurately now with our technology. So we really actually don't need that fossil. As a matter of fact, in a lot of museums, a lot of what you see are not actual fossils. They're just like reproductions of the original fossil. And even like in the Smithsonian, a lot of those really important fossils are in a basement in a drawer somewhere. What you're seeing as the fully articulated T-Rex is a bunch of plastic and technology that's not actually real. In some cases it is, right? But they don't generally tell you, it's like, oh, this is a plastic reproduction of T-Rex, because it's literally, I mean, they literally copied it from the original fossil. Not unlike if you've had a crown, for instance, right? Not unlike the way they do with the crown on your tooth, right? Where they basically take a mold of the original tooth, like an exact mold of your tooth, and they send it off to the lab to have an exact replica made of that, and then they kind of grind the tooth down and they put their cap on, right? It's exactly the same. It looks exactly the same, feels the same in your mouth. It's the same basic idea. So technically speaking, I would argue that fossils are not necessary. And of course, from my genetics, genomics background,
Yeah.
I've never been a fan of the fossil record. I've always thought it was a bit of a yawner, honestly. That's one of the reasons why, you know, I'm not a big phyletic gradualist, because that was their main exhibit A, is the fossil record. And I look at the fossil record, I'm like, okay, this isn't good enough. This is not good enough. I mean, it's interesting in some sense, but it's not good enough for what you're trying to do with it. And so genomics, on the other hand, is like, that opens it all up, right? So here's things that challenge our status quo, and that's good. That's what I mean by poking the bear, right? Poke the bear, poke status quo, turn it upside down. Those of us who are honest about what we're doing, that's a good process. That's a growing process, okay? The other one, of course, is genomics. All the information we're getting genetically from the human genome and species and things like that is enriching this entire story, okay? So rather than talking about fossils, I would rather talk about genomics, because fossils are like, yeah, so you got I guess it's not a rock in the dirt anymore, maybe. You got something in the dirt, great, good job. Bravo, why don't you talk to me about genomics? Yeah, so genomics is basically the study of your DNA architecture across an entire human perspective, or in this case, the human genome, for instance, or different organisms have their entire genome sequenced. So about 22 years ago, I don't know if you guys remember this, we sequenced the entire human genome. And it took a long time to do that. Now we could probably sequence any organism we want in short order. And the cool thing about that is, because evolution is fundamentally a genetic process, think about it. Darwin's paper is the transmutation of species. How do species transmutate? That is to say, change. You change your traits, you're changing your gene that governs that trait. So evolution is at heart, any species change is at heart a genetic conversation. So in order to understand how species are interrelated with each other and how they change, it requires you to understand genetics. Matter of fact, if you don't understand genetics, I would argue that you don't understand how species are related, which is a reason why I rephrase Dawkins' saying, "Do nothing in biology makes sense unless you view it through the lens of genetics." So now then, We've got genetics at its heart. That's a big idea. Evolution, obviously, changing species over time. That's a big idea. So there's a lot of big ideas in biology, and they all help each other if you allow them to. But what is all this for, right? I mean, think about this. This is great. This is all nice, but why, right? Why are we changing? Why are things changing? Why do we have genetics? What's the whole point? Where's this all going? Right? For those of you who like a destination, where's the destination? It's very simple. Let's ask this question. I want you guys to try to answer it. What is the ultimate purpose for every biological organism on this planet? The reason why every organism is here. Biologically, not psychologically or anything else. That's it. Done. Right? That's it. So think about it for a second. What does it take to reproduce? What do you need? Just think about us, right? In order to reproduce, what do we need? What? Okay, fair enough. You need two individuals. Yep. Okay, just think about just like the body, right? The organism body specifically. Like what organismically do we need in place? The organs, right? So we need the hardware to do so, right? The males need reproductive organs that does their job. The females need theirs as well, right? So they need all their reproductive organs. They need a few extra things, of course, in their body, since they will be carrying the offspring, right? So they need also the uterus in order to actually hang on to the offspring as well. So here's a good question. That, in order to get your reproductive organs, is something that you don't have right off, do you? We all start off our life as what? Single cell. Yep. One cell, which is a zygote, right? And so do you have reproductive organs at that point? No. What do you have to do in order to get those? Grow and multicellular, which is these two. Development, which is the creation of structure in the first place. Growth, which is getting that structure to be bigger, right? So you need to grow and develop. When do you start that process? Meaning what? Upon conception, right? As soon as basically, you're exactly right. As soon as fertilization happens, you're already starting this process.
The cells split.
Mm-hmm. And upon fertilization, you go through your first cell division, like almost immediately, as soon as the father's pronucleus enters into mom's egg. So that split happens immediately, like within seconds. It's very quick. So that happens. Now, here's the thing. As you're growing and you're splitting and things like that, you're becoming multicellular. Are you ready to reproduce yet? No. Okay, let's kind of fast forward 9 months. What happens then? Yeah. At birth, what do you have? Do you have all your parts? So you have reproductive organs. Yep, they're there, right? Males have fully descended testes. Females have uterus, vagina, everything. They've got all of their eggs, actually, that they will ever have in their life at birth. So they've got everything. The only thing that's actually really kind of developing, two systems that are majorly developing post-birth is the lungs. So they kind of have to develop a little bit. And much more so is the nervous system, right? That's also our brain and our nervous system is developing. So babies are largely just little bags of reflexes when they're first born. So they're still learning, you know, all their circuitry is still kind of being put together. That's one of the reasons why we don't have a lot of memories before the age of 4 or 5 or so, is because during that time, the brain is developing and it hasn't really had a chance to hold on to memories just yet. That starts to get to a level of development where you can hang on to that a little bit better around the age of 5-ish or so, give or take a year. So you're not ready to reproduce, but you have the parts. So what else do you need? So you need to get what? When are you able, bodily, physically able to reproduce? Yeah, puberty, right? That's about when it starts. That's when the males get their rush of testosterone from the testes. That's the reason why they turn crazy. Trust me, I remember what it's like to be a teenage boy. And now that I've got 2 daughters, I'm scared as hell.
Are they teenagers?
Not yet, no. One is 10, but she's starting to get to that point where she's getting close. Yeah, so I'm like, oh, crap. I gotta go shop for a shotgun. I don't know what to get. Single barrel, double barrel. I don't know. Maybe crossbow, right? I could go crossbow. That'd be a little more sporting. I just say like, listen, you got 5-second head start, go.
Try and swerve, maybe you can.
Yeah, right. Don't wear anything red. I'm kidding, but not really. But yeah, I mean, so are you at the age of puberty, right? Are you ready to reproduce? Physically, yes, right? Is it a good idea to reproduce? Why not?
Isn't it biologically the something you are able to produce? If you have a baby, they're not either you're not more trouble during the birthing.
That's good.
Less good.
More problematic, more of a high risk pregnancy if you're younger. Okay, I can go for that. But still, we've got technology. We can make it work. Why not?
It's not ethical, is what someone would say.
Okay, so ethical, that's kind of getting into a different realm. That's kind of us, right? But from a biological perspective,
Oh.
why would you not have a puberty like straight out of the gate getting pregnant? Your body is still developing. That is true. And that's going to change the whole thing. Anybody who's gone through pregnancy, it is like a physiological maelstrom. I mean, it turns the female body into another creature. I mean, it's amazing physiologically what the female body goes through during pregnancy. So yes, it will have some titanic consequences on the female body. Think about it this way. If you wanted to propagate the species, is it a good strategy to have all of your 12-year-olds have kids? Yeah, and what's the problem with that? From a biological point of view.
They're not mature enough, so they can't learn.
So why would they learn? What are they learning?
Survival.
Ah, bingo, right? That's where the babies having babies thing is, right? The reason why you don't have babies having babies is because a baby who's having a baby doesn't have the maturity and the development enough to actually defend themselves, much less their baby. So really the lion is thinking, hey, buy one, get one. Right? So let's fast forward to like 18. Better prospects?
More.
Of course, it depends on who you are, right? There's some 18-year-olds who should not be having kids. There are some who are like, wow, you are super wise beyond your years. You're going to be, legit. But 18 certainly is better than the 12-year-old, yes? Why? Because the 18-year-old has had the maturity and the experience of seeing their mom or dad fight off the life. So they are now better equipped because they've learned how to take care of their baby. So by the time they're 18, they've seen it happen. They know how to defend and survive, and they know how to defend and survive themselves and their baby. So survival is a big thing. So if you can't survive, is it any good? You're wasting your time, right? Because if you can't live long enough to reproduce, guess what? That's a fail from a biological standpoint. Okay, what does it take to survive? Everything else, right? Everything else on this list all points toward where? Reproduction. Let's think about it. Homeostasis is what? It's the process of maintaining a constant environment inside your body so that you can actually do physiological work. You need that in order to grow and develop, which you need to do in order to reproduce. Now, that's great. But as you're growing and developing, guess what you also need? You need organization, don't you? You need to be multicellular. So this patch of cells needs to go over here. This patch of cells need to go over here. You need to become brain tissue. You need to become skin. Another patch of cells becomes heart. So you need to divide up labor. You need to organize your activities. Development is the most bewildering and beautiful series of organized developmental events. It's just mind blowing. I mean, to go from nothing to this thing and what it has to go through and everything has to be exactly right is a feat of absolute mind blowing complexity and beauty that there's nothing like it on the planet. But you have to do all that, don't you? Because you have to organize yourself in order to what? Survive long enough to grow and develop, which will help you survive. And then if you last long enough to grow and develop, then you can reproduce and you're successful. But guess what it all takes? Nothing is for free. So you need to have an energy plan. So this will power everything in the body. You need structure to grow. Guess what? You need to pay for it. Energy. You need to develop a process in order to get whatever you, guess what? You need to pay for it. You need to reproduce. Guess what? You need to pay for it. You need to organize your tissues into all of the anatomy and physiology that we have now. Guess what? You got to pay for it. Every cell you build, every process you create, you have to pay for it. So you only get as far as the amount of money you have. So your energy plan is basically the foundation upon which you will build your living organism. And every single organism has to answer these big biological questions of life, including heredity, which is taking your reproduction and passing your traits on to the next generation. That's genetics. All of this has to be answered. Every living organism on the face of the planet has to answer these questions. They have to come up with some sort of a strategy to do all this. But guess what? They don't have to all do it the same way. They can do it however they wish, as long as they answer the question. So it doesn't matter how you do it, doesn't matter how you get there. As long as you get to a place where you can reproduce, you're successful. There's a lot of ways to do it. That is where our diversity comes from. We all answer the same questions, but we all have different ways of answering it. And you'll hear me refer to these big biological questions of life throughout the semester. Okay, so let me stop there. And then what I'll do is I'll use the next couple of slides to kind of do a quick, like a refresher, like an intro, say like, oh yeah, by the way, remember we did this. That'll be on Wednesday. And then we'll kind of move through and finish up our chapter. No, the homeworks that are under the quiz section are typically wired into exam dates. So exam 1 should be more like week 4 or so. So in a couple of weeks.